Probably yes. And thank you. I think we're working with your president. So I think it's good that you do it, right? Right. So yeah, we have 2 minutes and then I think we're ready to go. Yeah, that's a little bit, right?
Aloha and welcome to day 2 of the APA JPS CAP IAP 2024 meeting. We're thrilled to continue this monumental gathering in beautiful Hawaii, uniting the global pancreatology community. Today promises another exciting lineup featuring original research presentations and prestigious Fran Brooks and Ishio Hongo Symposium. Tonight's highlights, don't miss the 2024 award reception and dinner. If you don't have a ticket in the back of your name badge, but would like to attend, please stop by the registration desk to check availability. Connect with our industry partners. Take time during breaks and exhibit hall hours to engage with our industry partners and learn about the latest advancements in pancreatology. Special thanks to our top sponsors, Pfizer, AbbVie, Kirkland, Ipsen Biopharma, Arrowhead Pharmaceuticals, Hitchburg Foundation for Pancreatic Cancer Research, Digestive Care Inc., Nestle Health Science, U.S. Pharmaceuticals. And don't forget to explore the posters galleries. Be sure to visit and take a closer look during the lunch break from 12:15 and 2:15. Mahalo for joining us, and together let's make the most of this incredible opportunity to share knowledge, build connections, and advance the field of pancreatology. And with that, my name is Sobeida Cruz Monserrate. I'm co-moderating this session on abstract session 1, just to make sure we're all in the correct room for pancreas cancer with my co-moderator here.
Yeah, good morning, everyone. My name is Marco Del Chiaro, and I work at University of Colorado. And I will start immediately with our speaker. I remember, everyone, you have 7 minutes for presentation and 3 for discussion. And I welcome here on the podium a good friend of mine, Professor Robert Grusman from Erlangen, Germany, speaking about chemoradiation therapy versus chemotherapy alone following induction chemotherapy for unresectable pancreatic cancer, result of randomized phase 3 trial, CONCO 007 trial.
Good morning, everybody. Good morning. Thanks a lot for the kind invitation. I would like to speak about our prospective randomized trial about neoadjuvant treatment. I have nothing to disclose. So to start with, you probably know the resection criteria from the surgical status, and you know that we divide resectable, borderline resectable, and locally advanced irresectable. And the aim of our study was to look into local advanced irresectable cases. So these cases are usually up to date, as standard of care is palliative treatment. So here's a clinical case to start with. Typical case you probably know was externally operated, resectable with infiltrating the mesenteric root. You will see the pictures here. All the vessels, venous and arterious, are in the tumor. So the patient came to us, was included into our study with FOLFIRINOX and then chemoradiation, and then the Whipple procedure with SMV resection, right hemicolectomy. There was no need for arterial resection. And the result you can see here, histology was T2 in 0, R0. So, and this lady still lives with good quality and without signs of recurrence. Then this is our study design. Some years ago, so 9 years ago, we started this study, and the most intelligent thing of this study was that you could decide whether you start with chemotherapy with FOLFIRINOX or even with gemcitabine, then after induction chemotherapy, CT again, and then randomization to radiochemotherapy or chemotherapy alone. And then we tried to restage the patient and, if possible, to go for operation. So prospective randomized style. The primary endpoint was R0 resection, secondary endpoints overall survival, resection rate, and survival after resection. So the first calculation was more than 800 patients. After an interim analysis with a low recruiting rate, we changed the primary endpoint in R0 with 525 patients. So this is the consort diagram you see here. We randomized over 300 patients, and from both groups, 60 people have been operated. And here are the characteristics of the patient in the two groups. There's no difference. And you see here the toxicity. There is a increased toxicity grade 3 and 4, mainly leukopenia and thrombopenia. Very important that we not only used R0 definition, but also CRM status, narrow and wide. This is important for the final results. So R0 wide is CRM negative with more than 1 millimeter distance between the resection border and the tumor. So to come to the result, overall, the study is negative. So there is no difference between radiochemotherapy and chemotherapy. Here you see the survival. It's for these kind of patients, locally advanced irresectable, not too bad, with 14 and 15 months median overall survival. But if it's getting much more interesting for those patients who got resected, if you go for all patients, of course, the survival is much better if you could resect this. You see here the difference in the median overall survival, 13 and 19 months, and this is also significant. But even better, if you look into the of course, not too big group of CRM negative patients, 44 patients. And here with locally advanced irresectable patients, we have in a prospective trial, median overall survival 33 months and 5-year survival rate more than 30 months. So this is very important, I would say. And you see also in the surviving curves that you have approximately 10% of patients with real long survival, even longer than 5 or 6 years. So how often has been resected? So we learned a lot at the beginning. We were quite conservative. We explored the patient, but not resected them. But in the last 3 years, from 2019 to 21, there was a resection rate of 30%. So this is something we educated ourselves in our German centers. And here you see a complication rate was not really different, only in grade 2. There was a significant difference, but this does not mean very much for the patient. You see here the distant metastasis was not influenced by radiochemotherapy, but the local recurrence. So the local recurrence rate is less common after radiochemotherapy, but this makes sense. So I can summarize, radiochemotherapy improves quality of resection, so meaning less local recurrence. Survival is equal, but with R0 resection, you have a 5-year survival rate of 18%, with a median survival of 2 years. With CRM negative resection, so white R0, median survival is 33 months, and the local recurrence rate is lower after radiochemotherapy. So I think in some cases, neoadjuvant treatment with radiochemotherapy has advantages, especially in these arterial advanced cases. I'd like to acknowledge all the German centers who took part in this really demanding study, which we are now going to publish. It's in review. And I also thank my team from Erlangen. Thank you very much, and I'm happy to take your questions.
Okay, we have 3 minutes for question. I have several, but I want to open to the floor. I mean, the question. Yes.
So Robert, we all congratulate you. Great study and very honestly presented. Just a simple question. You showed after the induction chemotherapy, a small proportion of patients went forward for resection. What kind of criteria did you use to say patient is resectable after induction chemo? So FDG PET, CA19-9, that kind of stuff.
Okay, that's a very good question. Resectability is always tough in these cases. So we had a blinded evaluation with 4 independent pancreatic surgeons, including myself. But every hospital could decide on his own. So in my hospital, we usually go for a resection if the CA19-9 at least goes not up, better it goes down. No signs of metastasis, so we always do MRI. We don't believe in PET-CT.
We have time for another question.
Very nice study. Couple of questions as far as granularity of the data, looking at biomarkers. So like CA19-9, as you mentioned, did you see better responses in patients that had a reduction in CA19-9? Did you look at germline genetics with BRCA2, knowing we can see that sometimes in the radiation therapy responses, as well as lastly, the pathology? Have you looked at response? And I hate to say it, but have you asked your pathologist to look at response in those specimens?
Yeah, very good questions. To the first questions, no, we haven't done this yet. So this is translational research we are doing now. We routinely, there was a CA19-9, but we took blood from every patient and serum to follow up for follow-up studies. And we had a centralized pathology in Bochum, and she also looked to regression, of course.
25-second question, 25-second answer, please.
Congratulations, great work. Radiation equipment and radiation protocols were the same at all sites. And are you looking at biomarkers to differentiate the response and who doesn't?
Yeah, we haven't looked into biomarkers yet, and the equipment was fixed within the protocol. So there's a high standard of radiation therapy in Germany in these centers.
Thank you, Robert.
Thank you. Our next speaker is Dr. Alicia Fleming Martinez from Mayo Clinic, talking about KRAS G12D with SOC2 knockout generates atypical flat lesions, the precursor to cancer. It is my pleasure to announce that she's one of our young investigator awardees and that her abstract won the Best Abstract in Pancreatic Cancer, generously funded and supported by the Hirschberg Foundation. Congratulations. Looking forward to your talk.
Thank you. Yes, so I am Alicia Fleming Martinez from the laboratory of Peter Storrs. Uh-oh, I broke it. There we go. Okay. So we have previously published that oxidative stress induces acidotuductal metaplasia, where we have macrophages which secrete cytokines such as TNF-alpha and CCL5. We also have oncogenic KRAS and TGF-alpha contributing to the generation of reactive oxygen species. And reactive oxygen species then leads to acidotuductal metaplasia. Okay, next. So SOD2 is downregulated in human and mouse low-grade pannin. So we looked at SOD2 because it's one of the key regulators of oxidative stress. Next. So we first looked at human acinar cells versus low-grade pannin lesions, and we saw that manganese SOD or SOD2 is downregulated in low-grade pannins compared to acinar cells. Next. And we also looked at our KC mouse model. So we have the P48 Cree KRAS G12D model. And in this model, we see that manganese SOD is expressed in our acinar cells, but is again downregulated in our low-grade pannin lesions. And that's quantified here. Next. So knockout of SOD2, that's what we did next. In this KC model, increases duct formation in ROS. Next. And so we were able to see that when we did an acinar cell isolation from our KRAS or KRAS G12D SOD2 knockout mice, and we embedded in collagen, we saw that with the KRAS G12D, we get some duct formation as expected, but with our SOD2 knockout, we get about a 4-fold increase in the number of ducts. Next. Additionally, we see that with the SOD2 knockout, we get an increase in ROS generation. Next. And then we looked at 4-HNE, which is a lipid peroxidation marker and a marker, of lipid peroxidation. So oxidative damage. And we saw that our KC SOD2 knockout mice had increased 4-HNE in their lesion areas. Next. So the next thing we wanted to look at is the type of lesions that we found in our KC versus KC SOD2 knockout. And we found that the knockout of SOD2 increased atypical flat lesions. Next. And so we looked at the abnormal tissue area. We saw about double the amount of abnormal tissue area in our KC SOD2 knockout. So these are about 14-week-old mice. And we saw that the majority of the lesions are low-grade pannin lesions. There are a few high-grade pannins in both groups, but we had about double the amount of low-grade pannins in our KC SOD2 knockout. And we noticed that there was another lesion type in these KC SOD2 knockout mice. So we consulted the literature and thought that they looked kind of like atypical flat lesions, but there are only a handful of papers on atypical flat lesions. So we contacted the expert, Irene Esposito, who has published most of these papers. So she is a pathologist. Next. Um, and we got her to look through all of our slides, and she was able to identify for us the atypical flat lesions that we were seeing. So you can see here how different these lesions look from low-grade and high-grade Pannin lesions. And we found that the KC-SOD2 knockouts had many more atypical flat lesions compared to our KC mice. So because they were so rare in our KC mice, we hadn't really identified them previously. But with this model, we were able to identify those very easily. Next. So what are atypical flat lesions? They arise from acinar cells and they are a direct precursor to tumor. So it's kind of an alternate pathway from the acinar to ADM to Pannin to tumor pathway. So it's another type of precursor lesion. Next. So atypical flat lesions arise from acinar cells. They lack mucin, and they are a direct precursor to PDA. Next. And so you can see just how different they look from the low-grade Pannin lesions and how different the stroma around them looks. So you can see here, there's really not much collagen around our atypical flat lesions. So we did a trichrome staining. So here we have collagen in blue, and we quantified the amount of collagen that was around the Pannin lesions and the atypical flat lesions, and we saw a marked decrease in collagen around our atypical flat lesions. Next. And so we wanted to further characterize these lesions since there's not very much published on them, and we found that they have less oxidative damage and less senescence. Next. So again, we looked at 4-H&E, our lipid peroxidation marker, and we found that the atypical flat lesions had less 4-H&E, which is kind of surprising because we know that with our KC-SOD2 knockout, we have more reactive oxygen species, we have more abnormal tissue area. So this suggests that our atypical flat lesions have some type of compensatory mechanism to deal with the oxidative damage and thrive in this environment. Next. So, what we looked at was a variety of antioxidants. We looked at NRF1, which is a transcription factor which controls other antioxidant genes. We found that this was up in atypical flat lesions. Next. And one of the target genes of NRF1 is GPX4, and GPX4 kind of stops the propagation of lipid peroxidation. And so we found that GPX4 is up in our atypical flat lesions compared to our Pannins as well. Next. And so given that ROS is linked to senescence, we next wanted to look at beta-galactosidase. And so we compared beta-galactosidase in low-grade Pannin versus flat lesions, and again found that the flat lesions had much less beta-gal, indicating less senescence. Next. And so the next point we wanted to make was to look at humane endpoint. So do our KC-SOD2 knockout mice reach a tumor endpoint faster? And we hypothesized that they would, since we had more abnormal tissue area very early on. The mice were really progressing at that early time point. Next. However, we didn't see any difference. Here we separated our mice between males and females to look at if there were any sex differences. We didn't see any sex differences, and we didn't see any differences in our KC and KC-SOD2 knockout mice when we looked at a humane endpoint by tumor. So we were thinking that potentially these mice need an additional hit to progress to tumor, potentially more inflammation. Next. And so we looked at a cerulin model. So we gave 3 rounds of cerulin and then evaluated our mice for a humane endpoint. And we found that the KC-SOD2 knockout mice had reached a humane endpoint much more quickly at a median of 37 weeks compared to a median of 81 for the KC mice. So with that additional inflammatory hit, the KC-SOD2 knockout mice reached a humane endpoint much more quickly. Next. And with that, I would love to take questions. I'd like to thank everybody in the Storrs Laboratory for all of their support and the pathologist that we consulted, Irene Esposito, as well as our histologist, Brandy Edenfield. Thank you.
Thank you very much. We have time for 1 or 2 questions.
Very nice mouse model. Out of curiosity, how does this reflect on the human correlate? Where does the gene itself and as far as the atypical flat lesions in the pathogenesis of pancreatic cancer in humans, where does that fall?
Yeah, so there's not very much published. There's one case study showing an atypical flat lesion having the tumor kind of growing out of that, and so it was hypothesized that the flat lesion was the lesion of origin in that particular patient. And then for SOD2, there are some patients who have a SOD2 polymorphism, and it results in less SOD2 at the mitochondria, and those patients have an increased risk of pancreatic cancer. Mhm.
Richard.
Richard Waldron from Cedars-Sinai. This was a really nice talk. I really enjoy the description of the AFLs and your findings. So I just wanted to clarify, first clarify, and then a brief question. The clarification, you said the AFLs had less collagen than the pannin. But in my recollection, they would have a lot more of the sort of CAFs or activated stellate cells, SMA positive cells are like nesting in these AFLs. Isn't that correct?
So we see kind of a variety of this. Overall, in our mouse model, we don't see any differences in SMA between our KC and KC-SOD2 knockout, looking specifically at the flat lesions. We see some areas where there are SMA positive cells and some where there aren't. So
They're quite clustered in my experience as well.
Okay.
So my question is regarding the role of the ROS.
Mhm.
You showed us they reach a humane endpoint faster, and that is under conditions where presumably the ROS are out of control.
Yeah.
So what is the sort of feeling about the mechanism? Is it accelerating mutation, or what other pathways can you come up with to explain that? Thank you.
Yeah, so we're still trying to understand what pathways may be at play here. For the flat lesions, we really don't understand much about the signaling. So one of the things that's kind of on the list is that we could do like a microdissection of low-grade pannins versus flat lesions and kind of look at what the different signaling pathways are and what makes those flat lesions so different from the pannins.
Maybe it's oxidative stress mediated PKD activation.
Quick question, please.
Hi, Jonathan from Belgium. So a really nice phenotype. I was just wondering if you looked at the how the acinar cells are plastic, which transcription factors they might overexpress, or how different they are compared to pannins to have these differentiated cell types.
We haven't looked at that yet. Yeah, I really don't have an answer for that. Sorry.
Thank you.
Well, thank you very much. Great talk.
We'll move for the next presentation, a prospective multi-institutional study on the impact of combining RNA analysis with DNA-based next-generation sequencing on the diagnosis of pancreatic cyst and identification of the advanced neoplasia by Dr. Helen Afghani from Johns Hopkins University. I'm pleased to announce that Dr. Afghani is a winner of the Best Abstract in Pancreatic Cancer, journalist founded and supported by Hirschberg Foundation. Thank you.
Thank you. So thank you for the organizers for the invitation to present our work today. I have no disclosures. So pancreatic cysts are highly prevalent incidental findings. They're classified into either benign, pre-malignant, or malignant lesions. More than half are actually considered pre-malignant with a variable risk of malignancy. There's different managements depending on the cyst type. In cross-sectional imaging and cyst fluid analysis for CEA and cytology, which have been standard of care, has limited success in classifying cysts and does not offer prognostic value. So over the last decade, there's been a lot of work that has shown that different pancreatic cysts harbor different DNA mutations. For example, VHL mutation is highly specific for serous adenoma, which is a benign cyst. And the presence of a KRAS, GNAS, and or RNF43 mutation is seen in IPMNs, which are pre-malignant cysts. We recently published a systematic review and meta-analysis looking at the presence of KRAS and or GNAS mutation in comparison to the performance of an elevated CEA, and we found that KRAS and or GNAS was performed much better than an elevated CEA in identifying mucinous or premalignant cysts. However, we also see that there are similarities and mutations across the cyst types. This figure shows the molecular progression model from normal pancreatic tissue to pancreatic ductal adenocarcinoma. And as you can see, there's accumulation of various mutations, specifically CDK N2A, TP53, SMAD4, PTEN, and a PIK3CA mutation. Pancreas-seq initially developed at a UPMC. Dr. Singhi, who is spearheading this project, it was a 22-gene DNA-based next-generation sequencing panel. It was prospectively looked at in 1,900 EUS FNA specimens across 31 institutions. And findings were that it's 90% sensitivity and 100% specificity for detecting a mucinous cyst, and an 89% sensitivity and a 98% specificity for advanced neoplasia. So this was taken into consideration with the new Kyoto guidelines that was published earlier this year, where they've actually commented on using molecular markers and being able to differentiate pancreatic cysts, but there's still a lot of room for progress. RNA-based markers have been used, more specifically PRK, AC, A and B fusion genes have been identified in 100% with 100% specificity for IOPNs, which universally harbor high-grade dysplasia at the least and 30% cancer. So the aim of our study was to evaluate the performance of DNA and RNA next-generation sequencing panel onto clinical assessment of pancreatic cyst fluid in a prospective multi-institutional study. So Pancreas-seq GC is a 74-gene DNA and RNA targeted panel, which includes DNA alterations, copy number alterations, gene fusions, and gene expression changes. It's also an integrative analysis to include the scoring systems for cystic precursors and detect high-grade dysplasia and associated cancer. And along with neuroendocrine tumors, it also risk stratifies PANETs as well. So our RNA-based pancreatic cyst testing, we use CCAM, which is an RNA version of CEA. And in this prior study published about a year ago, it showed that CCAM5 actually performed better than CEA for mucinous cysts. So Pancreas-seq GC is again a 74 gene DNA RNA-based panel. We prospectively looked at this in about 4,200 EUS cyst fluid specimens across 47 countries. What we found is that a majority of the pancreatic cysts did harbor the defining genomic alteration. So for example, KRAS and GNAS were seen in IPMNs, VHLs for serous adenomas, MEN1 for PANETs. But we also saw that there's a subset that exhibited ambiguous results. For example, there was a presence of MEN1 and a VHL mutation, making us clinically question whether this is a PANET or a serous adenoma, or the presence of VHL mutation with the KRAS and GNAS mutation, making us wonder, is this a serous adenoma or a mucinous cyst? So based on DNA alterations alone, not all mutation-positive cyst specimens could be classified. However, with the combination of the DNA and RNA-based testing, we were able to better classify multiple cyst types. So an expression of a CCAM5 was used for identifying mucinous cysts, especially in those that were KRAS or GNAS wild type. PRK fusion genes was used for IOPN with at least high-grade dysplasia, and CHGA for PANETs, especially those that were VHL mutant or RNF43 mutant. So this slide is going to summarize the rest of our results. So there was 95% sensitivity and 98% specificity for mucin assist, 95% sensitivity and a 97% sensitivity for advanced neoplasia. There's a 100% sensitivity and specificity for IOPNs, and a 100% sensitivity and specificity for CHGAs and identifying neuroendocrine tumors. So in conclusion, the addition of RNA testing to DNA next generation sequencing improves molecular assessment of patients with pancreatic cysts, especially in identification, determination of malignant progression, and in cystic PNITs. We're currently accruing more follow-up data, which would add to the follow-ups, which would add to our data. So I'd like to acknowledge, again, Dr. Singhi, who spearheaded this, as well as the group at UPMC and all of our collaborators. Thank you.
Thank you very much. We are a little bit behind the schedule, so one question. Who has a question? Otherwise, I have a question. Okay, I have a question. So I think one of the next challenges would be to try to identify markers specifically for high-grade dysplasia. You know, most of the study we have today are putting together cancer and high-grade dysplasia, which is a problem because, I mean, cancer is a failure of the screening and high-grade dysplasia is really the target. Do you have any comment about that?
Well, we think, and again, correct me if I'm wrong, that the presence of some of these SMAD4, TP53, P10, and CDKN2A may actually indicate that it's not yet cancer and that it's just moving towards cancer. So hopefully we'll be able to detect high-grade dysplasia. But you're absolutely right that a lot of them are just clumped together.
Thank you very much. And again, congratulations for the award.
Thank you so much. Moving along, our next speaker, Xin-Shi Wang from Peking Union Medical College in China, title NGAL induced by IL-17 facilitate the formation of neutrophil entrapment traps, extracellular traps, and the development of immunosuppressive microenvironment in pancreatic cancer. Thank you.
For introduction. Good morning, everyone. I'm Xianzi Wang from PUMCH Beijing, China, and today I'm going to report our recent research on the IL-17-induced NGL facilitate the formation of neutrophil extracellular traps and the progression of pancreatic cancer. So the previous study had indicated that IL-17 play a very important role during the tumorigenesis and TME of PDAC. However, although the evidence in lab support this phenomenon, real progress in the clinical trial is rare. So to solve this problem, we first of all overexpressed IL-17 in a KPC mouse by using the AAV. And after 5 weeks of injection, we observed the overexpression of IL-17 in the acinar cells in the KPC mice. And also we observed a greater area of the pending and tumor area in our 17 treated group comparing to the control group. And next, we took an overview of the TME of the tissue, and we found that the neutrophil infiltration is significantly increased in the IL-17 group. And by doing a more detailed subgroup, we can see that the infiltration of IL-17, the infiltration of the neutrophil can not only be explained by the disease course of PDAC, but also be explained by the IL-17 in the presence of tumorigenic mutations. And next, we try to figure out how the IL-17 influenced the function of the neutrophil. So we treated the neutrophil with the condition medium of KPC plus IL-17 later, and the condition medium of KPC incubated with IL-17. And we found that the neutrophil was activated and the ROS production got improved and the migration ability got improved by the IL-17 condition medium, but not the IL-17 itself. And next, we performed the RNA-seq to the KPC cell incubated by IL-17, and we can see many of the downstream pathways got activated. And meanwhile, many of the cytokines and chemokines, including the NGL, 601, 602, got overexpressed. And next, we try to narrow down which is the target, the most important protein for the further analysis. And we can see that the relative amount as well as the absolute amount of the NGL got overexpressed significantly. And meanwhile, the NGL expression can be promoted or by the IL-17 in many other PDX cell lines, including the BXPC3, PANC1, SPC, and so on. In addition, we also observed the DEGs induced by IL-17, and we observed that many of the DEGs were NAS related. And considering that the ROS production got also promoted, we reviewed our mouse model, and we found that the NETosis in the IL-17 subgroup got promoted, indeed. So next, we try to figure out if the IL-17 facilitated NAS formation is NGL dependent. And to begin with, we verify the expression of the NGL receptor on the neutrophil. And then we treated the neutrophil with different conditions and took the PMA as the positive control. So when we add the KPC condition medium to the system, the NETosis just got a little bit improved. However, when we add IL-17 condition medium to the system, we can observe this dramatic increase of the NETosis. However, when we knock out the NGL in this, of KPC, the phenomena disappears. And when we add the recombinant NGL to the system, we got the phenomena recovered. So we can draw the conclusion that the NETosis induced by IL-17 is NGL dependent. And we all know there are two different kinds of NETosis. One is suicidal and another is a vital one. So we use the electron microscopic to observe the morphological changes during the process. We can see that the NAS induced by IL-17 condition medium share many similar morphological features compared to the PMA, which is suicidal NETosis. Well, next, we try to study the mechanism during this process. And based on the previous studies, we focus on two pathways. One is ROS production and another is autophage. And here we use the condition medium IL-17 as a positive group and took the inhibitor of P84 as the reference because P84 located in the very downstream of the pathway. When we use the inhibitor of the autophage, the chloroquine, the phenomenon of NETosis did not get infected. However, when we used three different inhibitor in a ROS pathway, we can see that the NETosis got impressed dramatically. And later, we try to figure out the upstream of ROS production. We used the IL-17 condition medium to treat the neutrophil, and we observed that the ERK, AKT, MAPK, and F-kappa-B got all activated. So which of the four pathways play a more important role here? We try to use different inhibitors, and we found that the ERK pathway might be more important during the process. Well, lastly, we try to study the function of NAS towards the KPC invasion, and we found that neutrophil itself cannot promote the invasion of the KPC cell, but the IL-17 condition medium. When we add the DNase 1 to the system, the promotion of the invasion got disappeared. One more interesting phenomenon is that we try to study the cell proliferation assay in vitro, but the proliferation did not get affected. However, when we build orthotopic model in vivo, the growth of the tumor can be accelerated by the NAS, and the phenomenon will be diminished in the presence of DNase 1. So to summarize of our first part of the study, IL-17 can induce the overexpression of NGL and activate the ROS and ERK in the neutrophils and promote suicidal NETosis. NAS can promote the tumor invasion, but can only promote the tumor growth in vivo. Thank you.
Any questions? If not, I have one. When you said you knock out NGAL to
NGL.
rescue, was it A whole body knockout or a conditional knockout?
It's in the cell line. Yeah, it's in the cell line. Thank you.
One real quick question. Did you happen to look at the Th17 cells in your mouse model?
You mean in the tissue?
Yeah.
We did, but sadly, there are very few Th17. You mean the source of the IL-17, right?
Exactly. So if you're not getting a lot of Th17s in
Yeah.
your tumor, where's the IL-17 coming from?
That is a very good question because we did a lot of experiments towards the tumor tissue of the mouse, and we haven't located the source of the IL-17. That is a very big problem. I don't know, maybe it's infiltration of the tumor of the immune cell being changed during the process of the PDAC.
Cool. Thanks.
Thank you.
Thank you very much. And I will move to the next presentation, mRNA translation, reprogramming in cancer and immunity. Dr. Kamini Singh from Montefiore Einstein Comprehensive Cancer Center.
Good morning, everyone. I would like to thank the organizers first to give me this opportunity to present our work here at the very nicest venue. This is my first time at Hawaii, so I'm enjoying this conference and the venue. So my lab works on mRNA translation, which is the process of making the proteins. We've heard so much about the genetics yesterday, genetics of pancreatitis and pancreatic cancer. And in my view, all these genetics, all the genes need to be converted into functional protein. And that's where the domain of mRNA translation stands. And that's what we are studying. And mRNA translation changes protein output. It can generate different protein forms. And based on what we know from these mechanisms, we can derive some therapeutic based on targeting translation. So for this talk, I would just want you to focus on two things. We use a technology called ribosome footprinting, which allows us to measure the changes in protein synthesis at the transcript level. So you get the whole genome-wide information. And we also can detect new protein forms and peptides and altered protein forms, which are coming from the same open reading frames, but it's not the same protein anymore because the open reading frame has changed. And we can detect those proteins and peptides and ask what they do. So coming to pancreatic cancer, we know that mutant KRAS is the driver of pancreatic cancer, and mutant KRAS signals to mRNA translation. There's a lot of signaling which we already know. I just try to put a simplistic diagram here, but all I want you to remember from this slide is that when you have activated KRAS, it can feed to mRNA translation directly to the cap binding complex, and protein synthesis can be altered. So we asked a very simple question that, are there any mutant KRAS specific mechanisms, or is it just feeding through PI3 kinase and mTOR, and whether mTOR is equal to KRAS or not? Those were the questions when I started this study. So we know that there are a lot of KRAS inhibitors, and that's what we try to use in this study. So we made use of G12C inhibitor and the G12D inhibitor, the Mirati compound and sotorasib, and we performed the ribosome footprinting. And in both the models, we can find out what are the mRNAs that are really sensitive to KRAS activity in a mutant KRAS specific manner. And we identified a subset of transcripts that are highly dependent on mutant KRAS activity. If we don't have mutant KRAS, then those mRNAs do not make any protein. So we first wanted to find out how RAS does it, what is the mechanism. So we can also map the ribosome coverage across the transcripts. These are complicated graphs maybe, but I can tell you the summary that when you inhibit KRAS, you actually block the translation initiation. So there is a big peak at the initiation site. You also start accumulating ribosomes upstream of the start site, and also downstream right before the stop codon. So suggesting that there is an initiation block and elongation block after you inhibit KRAS activity. So next we wanted to know what are the targets that are affected by KRAS. And then one big category of targets belongs to translation factors. We find almost every initiation factor and translation elongation factor, which includes all the many ribosomal proteins. So it's like directly affecting the ribosomal machinery itself. We validated all these targets in different models. I don't have time to show you, but I wanted to show you a very cool mechanism that we found but from this study, that RAS is regulating translation of mRNAs they have shorter UTR and they have somehow less folding or weaker secondary structures, which was kind of different from what we have learned from mTOR regulated translation and cap dependent translation. So this kind of summarized that what we know from this data. RAS is somehow feeding to translation initiation complex and elongation complex directly by changing the protein synthesis of these factors. And that's what we think that is controlling the downstream output of the translation. I did not have time to show you every data, but MYC and glycolysis and fatty acid oxidation, these are some of the other pathways that are also regulated by translation. So to summarize this one, this part, I think we somehow found a distinct mechanism. So mTOR is supposed to regulate all the translation, and top dependent and long UTRs, cap dependent complex. My previous work showed that EIF4A RNA helicase regulate many mRNAs that have long 5' UTR and complex structure, and they are probably hard to translate. And these are mostly oncogenes, metabolic proteins, and transcription factors. Many rest of the mRNA, that's what I call in this slide. But what KRAS does especially is regulating the translation machinery. And somehow that regulation is uncoupled because these mRNAs have short UTR and they have weak structure. So I can put them in a different bin that is regulated directly by KRAS. And that's where we think the mechanism is happening in a RAS specific manner. We also have data that I'm not going to show here, but RAS, when you inhibit RAS, TOR activation happens and 4A activation happens. There's a lot of feedback signaling that happens. Okay, so next, I just want to give you 3 more slides here showing that where we are going in future in the lab. We are trying to do to understand translation reprogramming during pancreatic cancer progression. And based on the data that we have right now, we have PANN samples, PDAC, and MET samples. And if you just look at the changes in the transcription versus translation during these different stage transition, you can appreciate that even at the early transition stage mRNA translation is perturbing a lot of genes. So it's much higher than the transcription. Transcription changes occur at a very late stage, I think. And then just a sneak peek, I'm not revealing all the data here, but we have identified unique proteins that are only made in one or the other stages of these pancreatic cancers, the normal versus PANN and PDAC and the liver mets. All these samples are matched. So PDACs were matched with the liver metastasis. So now we are investigating the role of these proteins as a potential biomarkers, neoantigens, and probably some therapeutics that can come out from this study. Thank you again, and please stay tuned, and I hope I can present this work in my next APA meeting. Okay, with this, I want to thank my lab members and all my collaborators for providing samples. And I mean, I especially thank my clinical collaborators, Christina Cabuzio and Michael Hollingsworth and Paul Grant-Gadget. I also want to thank Hirschberg Foundation because that was the first funding that we got for this project. And I'm very thankful for that because this project of finding neoantigens through translation and ribosomes is pretty exciting. And I hope we will find something very good there. Thank you. And I'll be happy to take any questions.
The paper is open for question. I think we have time for one question. Yep.
Elegant talk, elegant data. Congratulations. Last night we heard 4 great talks on KRAS, and yours could have easily fit into that.
Yes.
Is there any connection to the integrated stress response pathway in relation to this?
Yes, very good question. And there is a whole lot of data that we have found because RAS inhibition also leads to ribosome stalling. And we have looked into the integrated stress response, and it is not the classical integrated stress response that is activated. So it's not GCN2 and EIF2 alpha, but it is a different set of proteins that can sense the stalled ribosomes and induce a ribosome stalling stress. Yes.
Thank you very much, Dr. Singh.
Thank you. Our next presenter is Dr. Ken Sung. We're going to talk about the elevated lactylation promotes immunosuppressive tumor microenvironment and therapeutic resistance in pancreatic ductal adenocarcinoma, and also pointing out this is one of our young investigators grantees.
Good morning, everyone. It's my honor to have a presentation here. My name is Kang Sun, a PhD candidate from Zhejiang University. My topic is elevated protein lactylation promotes immunosuppressive microenvironment and therapeutic resistance in pancreatic cancer. Pancreatic cancer remains a significant challenge in clinical trial. Immunotherapy is considered promising for long-term survival. However, in a randomized control trial, CISPD3 conducted at our center, a combination use of immune checkpoint blockage can improve the overall response rate, but no clear survival benefit was observed. A significant proportion of pancreatic cancer patients are resistant to immunotherapy. Therefore, discover the pivotal drivers of immunotherapy resistance is key to enhance treatment outcome. Immune-cold pancreatic cancer is characterized as elevated glycolysis and resultant lactate. Whether elevated glycolysis promotes pancreatic immunotherapy resistance is not fully understood. It is well acknowledged that lactate influences the tumor microenvironment. However, whether spatial gradients in lactate distribution, lactate effect on spatial variants of cell, heterogeneous responses of immune cells to lactate not fully understood in pancreatic cancer. Recent studies have revealed that lactate can modify lines and residues on protein. In this research, we focused on lactylation to identify critical proteins and special sites for potential therapeutic intervention. We have found that high pan-lysine lactylation levels correlated with high level of macrophage infiltration, but low level of infiltration for CD8 T cells. We also found that elevated glycolysis level in tumor promote lactylation and the phenotype reprogramming of macrophage. We then discussed whether glycolysis and resultant lactylation contribute to immunotherapy resistance in pancreatic cancer. In our center, we conducted prospective cohort study that enrolled PDAC patients who received 18F FDG PET-CT before immunotherapy. And we collected biopsy specimens from patients enrolled in a randomized phase 2 CISPD3 trial and stand with pan-lysine lactylation antibodies. We have found that elevated lactylation is associated with worse survival in PDAC when treated with immunotherapy. It has also known that KRAS G12 mutation promotes glycolysis in cancer cells. We also found that KRAS inhibitor MRTX1133 decreased glycolysis and sensitized tumor to anti-PD-1 immunotherapy. We found that glycolysis-mediated immunosuppression was dependent on macrophage. We also found that glycolysis-dependent CCL2 secretion mediated macrophage infiltration. We also found that high CCL2 level correlated with worse survival and therapeutic resistance. To identify lactylated protein that regulate macrophage infiltration, we performed the multi-omics analysis to search for lactylated protein of both human and mouse pancreatic tumor samples. We found that tumor cell produced lactate induced K63 lactylation of endosulfin alpha, a crucial step that triggers stage 3 CCL2 signaling in tumor cells. Mechanically, ENSA K63 lactylation inactivated PP2A enhances SARC kinase activity and eventually increase stage 3 phosphorylation and boost CCL2 secretion. Lactate, mainly produced by tumor cells through glycolysis, is secreted into the TME as a signaling molecule. tumor-derived lactate upregulated TAM selectation and stage 3 phosphorylation. It is to say that elevated glycolysis not only increase infiltration of macrophage, but also promote lactation and pro-tumoral signature in macrophage, such as CCL2, arginase 1, S100A9, and IL-10. We finally discussed the transformation value of NSA stage 3 CCL2 axis. We found that NSA K63 lactation was positive correlation with stage 3 phosphorylation and was negatively correlated with overall survival. We also found that NSA K63 lactation blocking peptide and CCR2 inhibitor sensitized anti-PD-1 therapy in both humanized and mouse model. In summary, we found that elevated glycolysis in pancreatic cancer promotes NSA K63 lactation, a crucial step that triggers stage 3 CCL2 signaling and recruit tumor-associated macrophages. Tumor-derived lactate drives transcriptional reprogramming in TAMs by NSA K63 lactation, promoting an immunosuppressive environment. Targeting mutant KRAS, CCL2, or NSA K63 lactation enhances the efficiency of immune checkpoint blockage therapy. 18F FDG PET-CT pathological biopsy samples pan lysine activation expression and serum CCL2 level in patients who predict clinical outcomes. In all, I will acknowledge my mentor, Professor Tim Bo Liang and Xueli Bai, and Zhejiang Provincial Key Laboratory of Pancreatic Disease. This work was funded by National Science Foundation of China. I sincerely acknowledge CAP and APA for this chance of presentation. Thank you for being here. Thank you very much.
Thank you. The abstract is open for questions.
Okay, thank you really very much, Dan. And I think we can move to the next presenter. Thank you again. The next presenter will be Dr. Daisuke Hashimoto from Kansai Medical University, talking about neoadjuvant treatment with changing chemotherapy regimen according to CA19-9 level for resectable, borderline resectable pancreatic ductal adenocarcinoma.
Thank you, Chairman. I'm Daisuke Hashimoto from Kansai Medical University. Yeah.
If it doesn't work, you can ask to go next and they can move from there.
All right, so next, please. Yeah, I have no COI. Next, please. Yeah. So neoadjuvant treatment is now the standard treatment for RP duct in the high volume center in the world. And in Kansai Medical University, patients receive the neoadjuvant chemo shows significantly better overall survival, 39 months, compared with the upfront surgery patient, 23 months. Next, please. So recently, the concept of biological PR has been proposed. It means that the prognosis is poor in the patient with high biological markers such as CA19-9. Next, please. The objective of this study was to investigate the effect on survival due to the changing the chemotherapy regimen targeting CA19-9 regarding biological PR. Next, please. This is a single-center retrospective study in Kansai Medical University in Japan. In total, 283 patients with RP underwent neoadjuvant chemo were included. So anatomical resectable PDAC with CA19-9 higher than 150 was defined as biological in BR in this study. Next, please. Yep. This is a patient flow of this study. After the first line of chemotherapy, the group A achieved our CA19-9 criteria less than 150. And from group B to D, patients showed a higher CA19-9 after the first line in neoadjuvant chemo. And group B underwent surgery even though high CA19-9, and group C and D received a chemotherapy change to decrease the CA19-9. So group C achieved the CA19-9 less than 150 after the chemotherapy change, and group D, CA19-9 was still higher even after the chemotherapy change. Next, please. This is a clinical pathological characteristics of the patient with higher CA19-9 after first-line chemo, means group B, C, and D. And patient received a chemotherapy change, C and D, showed significantly lower CA19-9 compared with group B without the chemotherapy change. And the resection rate was lower, and CAP score was significantly better in the chemotherapy change group C and D. Next, please. This is a detail of the chemotherapy change in group C and D. Gemcitabine plus S1, this is a standard treatment in Japan in the first line, and gemcitabine plus nab-paclitaxel in the chemotherapy change were the most common regimen. Next, please. The CA19-9 response rate in the patient with chemotherapy change was 67%. It is significantly better than the patient without the chemotherapy change, 39%. Next, please. And the patient with chemotherapy change, C and D, showed significantly better overall survival, 36 months, compared with the patient without chemotherapy change, group D, 26 months. Similarly, progression-free survival was significantly better in the group C and D. Next, please. In the multivariate analysis for overall survival in the patient with higher CA19-9 after first-line chemo, the preoperative CA19-9 less than 150 and the chemotherapy change were independent prognostic factors. Next, please. This is the overall survival after induction of neoadjuvant chemo of each group stratified with chemotherapy change and preoperative CA19-9. The overall survival was 56 months in the group A without the chemotherapy change but achieved the CA19 criteria, and only 26 months in the group B. On the other hand, the group C, a patient received the chemotherapy change and achieved the CA19 criteria, showed better overall survival, 64 months. And group D, a patient received the chemotherapy change, but CA19-9 was still higher after the neoadjuvant chemo. OS was only 19 months. So we want to say that it means that the chemotherapy change improved the overall survival from green line to black line. Next, please. In the progression-free survival, in the group A, the PFS was 28 months, and group B, the PFS was only 12 months. However, in the group C, the PFS was improved to the 36 months. Next, please. So our study includes several limitations. One of that is this is only the single center retrospective study. And other is about the CA19-9 negative patient. However, we think we cannot distinguish the CA19-9 true negative patient with the false negative patient. Then the whole patient were included in our study. Next, please. So this is our recent treatment strategy for the PDAC. For the biological BR, mean the CA19-9 higher than 150 patient, now receive the gemcitabine plus nab-paclitaxel for the first line neoadjuvant chemo. So after that, CA19-9, if the CA19-9 does not respond, the regimen should be changed to the FOLFIRINOX or other regimen. Next, please. Yes, this is a conclusion. So also it need to be verified in the future study, including more patients and more centers, and changing the chemotherapy regimen targeting CA19 level can improve the outcome of the RBR PDAC patient. That's all. Thank you very much.
Thank you very much, Dr. Hashimoto. Is there any question from the audience? I do have a question. Oh, there is a question, please. We are able to take only one question for this paper.
For the interesting talk. I'm Jennifer Vallerin from UCI Medical Oncology. So it seems to me, just from looking at the differences between groups C and D, that it's not necessarily the chemotherapy change itself that's important. It's just the fact that you have a lower CA19-9. However it is that it's gotten there, because group A also seemed to do well regardless of the chemotherapy change. So isn't it just more important that you're dealing with distant disease or getting a patient to a point of resection and not necessarily how that's coming about? So isn't that still more just consistent with data that we already have with a low CA19-9, like particularly under 170 does better and should go to surgical resection versus others?
Oh, so you mean the chemotherapy CA19? So yeah, we want to say that the neoadjuvant chemo should be done according to the biological response or the markers. So we want to say that chemotherapy change is not so. We want to say that the CA19-9 targeting chemotherapy neoadjuvant chemo is important, but the chemotherapy change is not now. Yeah.
Because I
Now
guess A
we-
to D would have been the question there, right? If you ended up chemotherapy change lowering the CA19-9 and also having improvement despite the change in the CA19-9.
Yeah. So the chemotherapy, if the patient achieve the CA19 lower CA19-9 only after the first line, so the chemotherapy change should not be done. So patient should go the surgery.
Okay, thank you.
Thank you very much.
Thank you.
Thank you. Our next speaker, Dr. Prasad Gandaway from the University of Kansas Medical Center, is going to talk to us about Avastin interrupts the histaminergic neurons pancreatic cancer communication loop to inhibit pancreatic cancer progression under chronic stress conditions. So a young investigator awardee. Congratulations.
Aloha, everyone. Thank you so much for this opportunity and selecting this great venue. I hope all are enjoying discussing science in the nature. So everybody gave really good introduction about pancreatic cancer. So I'm going to start a little bit story, how I started this research. So when I started my lab 3 years ago, I was working with a patient advocate. She was pancreatic cancer survivor. She told me all I felt during my pancreatic cancer journey was stress. I was so stressed. That was one thing. Second thing was very interesting what she said. She said one of her friend, her husband died of colon cancer. And when she got detected with pancreatic cancer, she had poor survival. She died within 6 months. And she was thinking that stress has something to do with it. And it stuck in my mind because we have anti-anxiety medicines to treat and why it is not producing the effect what we're looking. So I started looking at this research. So nerves are the one which respond to the stress, and tumors have a lot of nerve innervations, especially pancreatic cancer. It has almost 40 to 60% pancreatic cancer shows extensive perineural invasion. So I started thinking like nerves are important. How nerves and cancer cell communicate, it's a very peculiar way. Cancer cells secrete neurotrophins, which attract nerves. And in the response to help from cancer cell, nerves secrete neurotransmitters, which binds to the receptors on cancer cell, and that helps for their growth and survival. So it's like symbiosis association which they are going on. I'm not the first one to think about it. There is one very good paper published in Cancer Cell from Dr. Wang's group, and they showed that chronic stress accelerate the tumor formation. And they use KRAS model. They gave stress for 6 months, and they start seeing pancreatic adenocarcinoma formation really quickly. And they discussed this norepinephrine binding to adrenergic beta-2 receptors, which activates CREB and NGF as a neurotropin. And very interesting paper. That I started thinking, okay, we already have a target, adrenergic beta-2 receptor. So can we just target it? But there are some problems. Adrenergic beta-2 receptor are present on the lungs, and its antagonism will result in bronchoconstriction. Second, we can use non-selective beta blockers, which will block beta-1 and beta-2, but beta-1 also present on the heart, and that it makes very complicated because if you antagonize beta-1 receptors on the heart, it might cause bradycardia or hypotension. So what are the possible solutions? Either you deliver adrenergic beta-2 blocker directly to the pancreas by using a drug delivery system, or identify new targets in this neural cancer communication. So for this, first I just went to TCGA database. I studied all the receptors which are involved in neural signaling, such as cholinergic, glutamate, adrenergic, serotonin, histamine, and dopamine. Out of all these, I saw surprisingly histamine receptor 1 is highly upregulated in all these receptors which are involved in neural signaling. So I started looking at histamine receptor. So I asked a question: is histamine receptor 1 expressed in pancreatic cancer cells and tissues? Before going there, let's see how histamine works. Histamine acts on histaminergic receptor, and then it activates gustatory and calcium signaling. This is a canonical pathway, but it also activates MAP kinase pathway and CAMP. So then next I looked if histamine receptor is upregulated in pancreatic cancer tissue, and here you will see that in cancer tissues, histamine receptor 1 expression is very high, significantly high. Similar data we found in cancer cell lines as well. So now we know that pancreatic histamine receptor 1 is overexpressed in pancreatic cancer tissues. The next question I ask is, like, from where histamine is coming? And there are few cells in the body which secrete histamine. First is definitely mast cells, which are well known. Pancreatic cancer cells also have mast cells. Then basophils, then histaminergic neurons. They're usually present in basal ganglia of the brain, and then enterochromaffin-like cells in stomach. And histamine is synthesized by an enzyme called histidine decarboxylase, which convert histidine into histamine. So I thought this is a good start because all of these cells will have HTC. I did this staining to start looking, and I found this staining when I went to the pathologist and showed him, like, hey, I was hoping to see mast cells, but I'm seeing this weird structure and I don't know what it is. He said, these are neurons, these are nerve fibers. And I was like, this is surprising because we don't know. This is the first time I've checked all the literature, and I did not see presence of histaminergic nerves in pancreatic cancer tumors. And that's where I started doing this research. So then I asked the question, can histamine activate this neural cancer crosstalk? And for this, I just take the pancreatic cancer cells, I treated them with histamine, and I looked at the same readouts with Dr. Wang's paper, like CREB and neurotropins like NGF, BDF. And this is the pathway which I was talking about. So when you treat cells with histamine, it increases or it activates phosphorylation of CREB in time-dependent manner, as well as BDNF and NGF, which are the neurotropins, which indicates that histamine activates this neural cancer communication through histaminergic neurotropin loop. Now, this was in vitro. Can we do this in vivo? And for that, I conducted orthotopic pancreatic cancer model. Now, chronic stress of immobilization, where we just take mouse restrainers, we put the mouse in it for 2 hours, and it induces chronic stress without causing the pain, is known to activate the neural signaling. And that's the same model we did. So this is the design of the model. For first 7 days, we just gave mice stress for 2 hours every day. On 7th day, we injected KPC cells orthotopically into the pancreas of the mouse, and then we continued giving this restraint stress up to 35 days. After 35 days, we euthanized the mice, and surprisingly, we saw this. The tumor growth after stress is at least 2 to 2.5 times faster, which indicates that chronic stress amplifies tumor growth. We also looked at neurotropins such as BDNF and NGF, which are significantly high after chronic stress. But surprisingly, we also saw histamine, which is going up. Then we wanted to see if this histamine receptor is playing any role in all this. And that's why we use shRNA to knock down histamine receptor. And then we conducted the same orthotopic model, same parameters. And here you can see black box is the control, red box is the stress. We see there is increase in stress response, but in blue and purple box, you can see the knockdown of the receptor completely rescued the effects of the chronic stress on tumor growth, BDNF, as well as histamine. So this indicates that, or this suggests that histamine receptor knockdown can break this neural cancer loop. Then the next question was, can we target histamine receptor by using antihistaminic drugs to interrupt this crosstalk? First thing we did, we just took all FDA-approved antihistaminics and ran the proliferation assay, and then we did orthotopic model. So these are the antihistamines which we tested. One of the antihistamines was Ebastin, which inhibited pancreatic cancer growth. If you remember the time course I showed before, where histamine is inducing BDNF and NGF expression, but if you treat cells with Ebastin and then treat with histamine, we don't see the activation, which suggests us that Ebastin incubation can rescue the histamine effects. Then the next thing we did was we ran the same tumor model, same way. Only thing, we treated Ebastin 60 minutes before we started the stress. We saw the same effects as we saw in HRS knockdown, where we saw with the stress, there is increase in tumor growth, BDNF and histamine, but when we treat with Ebastin, it actually rescues all the effects of the stress. So to summarize, chronic stress or anxiety activates histaminergic neurons. It induces neurological inflammation, releases substance P, which serves as a chemoattractant as well as mast cell degranulation for mast cells, which releases histamine, binds to histamine receptor 1, activates CREB and BDNF. But if you use Ebastin, it will break the loop to overcome the cancer growth. So these are my acknowledgements to my group. Dr. Hindol Ghosh has done most of the work. And thank you for the funding agencies.
Thank you very much. We have time for one question.
Did I go too fast?
Any epidemiological studies on the use of people that have used antihistamines and their development of cancer, pancreatic cancer in particular?
Yeah, so there are some studies which are published which talks about use of antihistaminics and less incidence of cancer, especially also there's one study in pancreatic cancer as well. Yeah, thank you.
Thank you very much.
Thank you very much. And I will call the last speaker, Dr. Yamazaki from Kyoto University Graduate School of Medicine, talking about intrapancreatic fat deposition as a novel risk factor for pancreatic ductal adenocarcinoma, a large-scale prospective cohort study.
Okay. Yeah. Thank you for the kind introduction, and thanks a lot for the organizers for giving me this opportunity to present our work here today. Yeah, so fat can accumulate within the pancreas as visualized by ultrasound and the CT. And histologically, pancreatic fat is mainly consisted of adipocyte within the pancreatic tissue. Pancreas fat can be quantified by CT or MRI, and the normal range of pancreatic fat level is less than 10%. And 1/5 of individuals have pancreas fat exceeding this 10% threshold. Importantly, randomized trials have shown that pancreas fat can be reduced through lifestyle modifications. But you may wonder that pancreas fat is merely an indicator of obesity, but it's not. These MRI images showed two individuals who have very similar BMI and very similar visceral fat content. However, the upper one has very low pancreas fat and the lower one has a lot of fat within the pancreas. Now, there's extensive research underway on the clinical impact of pancreas fat. We've been active in this area for a decade and showed that pancreas fat is a risk factor for type 2 diabetes through the paracrine effects to the beta cell function. Also, pancreas fat is considered as a risk factor for pancreatitis. I will be presenting a poster tomorrow, so please come to the poster session on this topic. Then one remaining question is whether pancreas fat increases the risk of pancreatic cancer. Proposed mechanism behind this is a secretion of inflammatory cytokines from the pancreatic adipocyte causing pancreatic cancer. Actually, there are several studies showing an association between pancreatic fat and pancreatic cancer. But these studies are cross-sectional or retrospective case control studies, and there is no prospective cohort study to address these important questions. Therefore, we conducted a prospective cohort study to investigate whether pancreas fat increases the risk of pancreatic cancer. So we conducted a large-scale prospective cohort study with a quite long follow-up period using UK Biobank, which is a biomedical database containing genetic, imaging, and lifestyle and clinical information from UK participants. In this study, we included 30,000 individuals who underwent MRI. Pancreas fat was measured via MRI with machine learning technique. Then using 10% cutoff, as I mentioned previously, the participants are divided into two groups, one high pancreatic fat group, and the other one is a low pancreatic fat group. Then participants are followed until 2022 for the incidence of pancreatic ductal adenocarcinoma, which was identified through cancer registry, hospital record, and cause of death data. Then we used a Cox regression model to adjust important confounders to assess the association between pancreas fat and pancreatic cancer. So let's come to the results. This table shows the baseline characteristics of the participants. The participants with high pancreatic fat group are older, included more male, and had a higher BMI, and they are followed about 5 years. And this figure shows the incidence of pancreatic cancer. Compared to low pancreas fat group, the participant with high pancreatic fat had 4 times higher risk of pancreatic cancer. So now it becomes really interesting. What about their pancreatic cancer risk after adjusting for important confounders? Here we adjusted for important confounders, and look, the participant with high pancreatic fat group had 3 times higher risk for developing a pancreatic cancer. But instead of binary categorization, we also use Tatar-defined categories. And still again, high pancreatic fat was associated with 3 times higher risk of pancreatic cancer. Then finally, we used a continuous value of pancreatic fat and showed similar trend. But it is important to note that the important limitation of this research question is reverse causation, where undetected pancreatic cancer could be causing pancreatic fat accumulation. To address this, we used another approach called Mendelian randomization, which uses GWAS data to investigate the association between genetically predicted pancreas fat and pancreatic cancer. Then, using this approach, we found that the participants with genetically higher risks, higher genetically predicted pancreas fat had a higher risk of pancreatic cancer, which means that genetic predisposition to the pancreas fat is also associated with pancreas cancer. So this supports our hypothesis that pancreas fat causing pancreas cancer, not the other way around. Our study is the first to show that pancreas fat is a modifiable novel risk factor. Therefore, reducing pancreas fat may decrease the risk of pancreatic cancer. However, it is important to note that besides adipocytes within the pancreas, fat can also accumulate within the acinar cells and endocrine cells. Current imaging modalities such as CT and MRI cannot determine specific location of the pancreas fat. So we need more comprehensive research integrating pathology, genetics, imaging, and clinical information. We thanks for the UK Biobank and the funding agencies. This project is a true collaboration across the globe, which is a lot of fun. I'm looking forward to your questions. Thank you.
Thank you very much. The paper is open for question. We have time for a couple of questions. Yes, please.
Thank you. It's great to see you, Marco. Wonderful and very hot topic, deservedly so. So we know in the liver that some folks accumulate fat and it's benign, and others it's associated with inflammation. Do you think the same is true in pancreas? And do you think imaging can detect the two?
Oh, thank you for your question, but I don't have any good answer for this. But at least imaging, as I told you, cannot determine where the fat accumulate. So now we need more pathological study where the lipid accumulate. The adipocyte or lipid in the acinar cells or endocrine cells, these may be different functional effect to the pancreatic cancer. Thank you.
Tim Antrikis, radiologist, and great study. Thank you very much for presenting this here. The pancreatic fat is definitely under examined in the pancreas. It is well known for the liver, but not for the pancreas yet. My question to you is, how did you determine the 10% rate? Currently, there is no, to my knowledge, there is no limit on what is low fat or what is the ratio of pancreatic steatosis. What was your guideline to say that let's call 10% as the limit for pancreatic steatosis?
Yeah, thank you for your question. Actually, but I think in 2014, there was a population-based study from Hong Kong, and they showed that excluding the patient with high alcohol consumption, excluding metabolic syndrome patient, then they determined that 95% total cutoff, which was 10.4%. So we use that cutoff.
So you used the study from Hong Kong.
Yeah.
Well, there is a study from Germany, which they did on the general population on more than 1,000 patients, and the German population has pancreatic fat somewhere from 2 to 6%. They never really go over 10%. And we did a study and published in the Indiana, our average rate was 12%. So it really depends. It's really hard to make a concrete judgment at this time. What is the level of pancreatic steatosis? The second thing is, I think that most of the pancreatic fat.
I think you should go, offline when the session is over. We are running out of time. It's very interesting topic. Maybe you can just, chat,
Yes.
later, if you don't mind. I want to be sure that we close on time as our responsibility. I want to thank my co-chair for chairing this session, all of you for amazing question, participation. And sorry, we need to cut a little bit, but we have that session coming. All the speakers, and congratulations again for the people that got the award. Thank you very much.